My name's Debbie Rees. I'm a biologist, and what I was hoping to do over the next 20 minutes is introduce you to what I know about the biology of ethylene, and give you a framework in which to understand how it works and how therefore the effects of scrubbing ethylene or using chemicals such as 1-MCP to block, or SmartFresh as you might know it, to block ethylene action. I'd just like to very briefly introduce you to where I come from. I work at the Produce Quality Centre. This is actually a storage research centre in the southeast of the UK. We work a lot on top fruit, but we also look at fruit that's imported to the UK. You know, there are many things that you can grow that we cannot. So in the UK, we rely on imported produce. Okay, so this is a brief summary of what I'm planning to cover today, an overview of the biology of ethylene. I want to introduce you to how ethylene works within cells, so to talk about the receptors and the implications of the way that ethylene works for how to manage it in terms of fruit ripening, rots, physiological disorders, and fruit flavor. So ethylene is a remarkable plant hormone. It regulates many biological processes in plants. So just some of them listed here. It regulates the break of seed dormancy. It regulates the initiation of flowering, the leaves falling off the trees in autumn, fruit ripening, senescence of green tissues. It's also a very important plant hormone for response to stress. So for crops to cope with flooding, to respond to physical damage, and to respond to rots. So as you'll see later how important this is, ethylene is really important for helping plant tissues to respond to rotting pathogens. I'm not going to go into lots of detail about the biochemistry, but just to give you an idea that we have sort of known that ethylene exists for a long time. Farmers have known that they can smoke orchards in order to initiate flowering. People have known that you can stimulate ripening of fruit with smoke. And this is all due to the fact that within smoke, there is ethylene and some other chemicals that mimic its action. Last century, a Korean, Shangfa Yang, actually spent a lot of his career working out the biochemical pathways for synthesis of ethylene. And don't worry, I'm not going to go into that. Okay, so moving on. What I want to do is to take you down into the structure of plant tissues. So here we have an apple, and apples are made up of cells. If you could basically expand that picture more, the cells, each individual cell is a very complex structure full of membranes. And if you go down further, the actual membranes around the cells and within the cells are themselves very complex structures with lots of large proteins embedded within that cell structure. So the receptors for ethylene, the way in which tissues respond to ethylene, are actually some of those biological structures, some of those proteins embedded in the cells. So here we have a much more simplistic picture of the edge of a cell. So ethylene, small little chemical, very volatile, binds to that receptor and turns it on. And once that receptor is turned on, it initiates a series of signals that make things happen in the cells. It basically turns on the nucleus to actually produce specific genes, to transcribe specific genes and start things happening. And one of those things that happens is fruit ripening. So this is initiated by those receptors within the cells across the tissues. So if we were actually to look at the structure of those receptors, this is actually a simplified diagram of what they look like. They are extraordinarily complex. But basically what happens is that in most of the time, the receptors are sitting there and the processes such as ripening are not happening. When ethylene comes to bind, it changes the structure of that receptor and ripening is triggered. Okay, so I did mention that ethylene controls many, many different processes, not just ripening. And the way in which the receptors control processes actually differ for different types of processes. And we talk about two different ways in which receptors work. We call them system 1 and system 2. So all those processes, most of the processes that I mentioned at the start, the germination of seeds, the control of flowering, the fall of leaves from the trees, they are all controlled by something called system 1. The ripening of fruit, however, is often controlled by system 2. So the difference between those two systems is actually quite important to you if you are fruit ripeners. So most of the time, you have a system where if you add ethylene, something happens. If you take ethylene away, that something stops. But for system 2, actually, if you add ethylene, you get a positive feedback and the tissues themselves produce a lot more ethylene. And what that means is that the system becomes unstoppable. So in terms of fruit ripening, we actually classify fruit in two different ways. We call them either climacteric or non-climacteric. Bananas and avocados, and in fact apples, are climacteric. And they go through a period of development and maturation in which all those systems are stimulated by ethylene, but using that system 1. They reach a point of ripening where they suddenly go to system 2. And at that point, they go into the climacteric. Non-climacteric fruits, such as strawberries and pineapples, are different, and they always stay in that system 1. So why am I even mentioning this? And I apologize if I seem to be going into more detail than I should. I just want to emphasize that these climacteric fruits, they differ from the non-climacteric fruit in that they have a ripening process in which a lot happens, and a lot happens very quickly. So I'm not sure who's in the room at the moment, whether there are banana ripeners or avocado ripeners, or whether you are involved in ripening. But if you are, then you'll be very familiar with that burst of activity that happens during climacteric ripening. Whereas the other fruits, such as pineapples and strawberries and oranges, they tend to develop in a much more even way. If we look at what happens for climacteric fruit during ripening, a lot happens at that point. There's a lot of respiration and a lot of ethylene production during that climacteric. What is important to people who manage fruit is that means it gives us an opportunity to control that ripening artificially by using this amazing chemical, ethylene, to trigger that ripening. So we use ripening rooms. We do not use ripening rooms for non-climacteric fruit. Okay. I just wanted to make a point, another point about ethylene, in that plant tissues can be extremely sensitive to the addition of ethylene. In ripening rooms, when we add ethylene, we are adding quite small concentrations. We talk about parts per million, and it's typical when we're ripening to use ethylene in concentrations of 10 to 100 parts per million. But some tissues are sensitive 1,000-fold lower, so to concentrations in parts per billion. And if there are any people in the room that manage kiwi fruit, it's particularly sensitive to ethylene, down to 5 or 10 parts per billion. Okay, so I wanted to emphasize that because the management of ethylene therefore becomes absolutely critical. Okay, so by the end of this short presentation, I will have gone through all the post-harvest biology that I teach my master's students in a year. So please bear with me on this. I'm now going to move on to SmartFresh, or 1-MCP, which is a chemical which we use to control the action of ethylene. So how does this work? Here is that picture of the receptor for ethylene that I showed earlier. SmartFresh, or 1-MCP, is actually, as far as the cell is concerned, rather similar to ethylene. It looks like ethylene. It also binds to that same receptor. But unlike ethylene, it doesn't turn that receptor on. It doesn't make all those things happen. It actually binds irreversibly to that receptor and keeps it in the off position. So that means permanently that receptor can no longer work and it can't respond to the addition of ethylene. In fact, the only way that that effect can be reversed is for new receptors to be synthesized, made by the tissues. That has quite important implications to the effect of 1-MCP for fruit, people who manage fruit. Okay, I'm going to do what I would describe as a sort of thought experiment with some bananas and try and show you what happens if you use ethylene scrubbing or you use 1-MCP. So one of the things that banana handlers get worried about, if they are sending bananas, for example, by sea freight to my country, the UK, they are sent green, and then when they arrive in my country, they are ripened. So what you want to do is avoid those bananas ripening in the voyage. So one problem that can arise is that if the banana has a rot, then it will start producing ethylene to try and protect itself against that rot. Unfortunately, for the shippers, that can cause all the other bananas in the consignment to ripen. It can trigger that climacteric ripening and cause all those bananas to ripen. Real problem when you arrive at the other end. So what can you do about that? Well, you could add an ethylene scrubber into the system in order to remove that ethylene that's being produced. So in that case, what you could do is you could actually protect the bananas theoretically nearby to banana B, but you can never protect banana B because it's producing its own ethylene, and that ethylene is diffusing through the tissues of the banana itself and causing it to ripen. Theoretically, if you used 1-MCP, which would go through all those bananas and block those receptors, if you got that in early enough, theoretically, you could stop all those bananas ripening. Okay, so there are all sorts of simplifications that I've just made. But basically what I'm trying to get across is the fact that there is a difference to using ethylene scrubbing and using something like 1-MCP. And why is this important? It's important because of those things that I said that ethylene does in addition to ripening fruit. But let's start with that ripening fruit situation. So in my country, we use 1-MCP widely to store apples. Admittedly, apples are usually stored after they've gone through the climacteric. We are aware that people sometimes use 1-MCP for other fruits such as pears and avocados in order to prevent ripening. However, once you've done that, you can get a problem because you can then get a situation where subsequently, when you want to add ethylene to ripen, it doesn't work. I'm just showing here a couple of slides of trials that we tried in the UK where we were looking at, for pears, whether it was possible to use 1-MCP and still be able to retain the ability to ripen the fruit subsequently. We tried it in two different ways. We tried using lower levels of 1-MCP than are commonly used, and we could get down to levels where we could subsequently ripen the fruit. We also tried something that might seem a bit wacky, in that we tried adding ethylene and 1-MCP at the same time. The rationale being that if that ethylene was binding to some of those receptors, it would block the 1-MCP, actually irreversibly blocking those sensors. And to some extent it works. It's not been tested commercially. I suspect it's too difficult a system to manage for it to work commercially, but it highlights that problem that one can have with 1-MCP and subsequent ripening. What about the fact that ethylene helps fruit protect itself against rotting? So this means that sometimes if you use 1-MCP to protect fruit against ethylene, you also turn off some of those protective mechanisms. So we have tried comparing the use of ethylene scrubbing and 1-MCP for long-term storage of Bramley apples in the UK. We've actually found that in that case, although both 1-MCP and ethylene slow down the softening and the color changes in that apple, actually the smart fresh fruit was more susceptible to rots than the scrubbed fruit. And we put that down to the fact that the ethylene, the fruit really needed the ethylene to help it protect itself against the rots. It was a very small effect, but it highlighted what was happening there. We are also aware that ethylene is important in protecting fruit against certain storage disorders. And again, forgive me, all the examples here are apple, but we can find out what ethylene is needed for by basically seeing what happens if we were add 1-MCP and block that action. So 1-MCP is very useful in reducing many storage disorders, but it's also, for some storage disorders, it makes them worse. And so, for example, carbon dioxide injury can be exacerbated by adding 1-MCP to certain apple varieties. And in the UK, one of our popular varieties, Braeburn, we never use 1-MCP for because we get those disorders. My colleague, Richard Colgan, who knows a lot more about the biology of ethylene than I do, did some trials where he was looking at the effect of both ethylene scrubbing and chemical AVG, which actually inhibits ethylene synthesis. So pre-harvest treatment of fruit with AVG. And in that case, he could see very clearly that by removing that ethylene, that he was increasing the disorders. I'll just make one final comment before I stop. and that's about flavor development. So during that ripening process that ethylene is orchestrating, is managing, there are several aspects of ripening in terms of texture, color, but also chemical composition and the chemical volatiles that affect flavor. So that isn't something we've worked on directly, but a quick look through the literature shows us that there are many reports where people have compared the flavor volatiles of fruit with 1-MCP and without 1-MCP. And in many cases, you can see that the number of volatiles, the concentration of volatiles decreases when you completely block ethylene. Thank you. I finish there.